In the previous session, we discussed about how acetyl coenzyme A is brought into the cytoplasm, how NADPH is available, and how CO2 is made available. We also discussed about the conversion of acetyl coenzyme A into the malonyl coenzyme A. In this session, we'll discuss about fatty acid synthase multi-enzyme complex. The objective of this session is, at the end of the session, the learner shall be able to explain fatty acid synthase multi-enzyme complex and its features. Okay. The fatty acid synthase multi-enzyme complex, it is a cytosolic enzyme. It is a cytosolic enzyme. Okay. It is homodimer. The enzyme is homodimer. Two similar units are present. It catalyzes formation of 16 carbon palmitic acid. It catalyzes formation of 16 carbon palmitic acid. Okay, palmitic acid is saturated fatty acid. Okay. As I said, two identical monomers are present. Two identical monomers are present. Okay. Each monomer contains 7 enzymes. Each monomer contains 7 enzymes and one protein. Okay. So that is acyl carrier protein. Okay. Then, so how the homodimers are arranged? So these monomers lie in head to tail orientation. They are arranged in head to tail orientation. Okay. Then the next one is, so let us see what are those enzymes and protein. We'll give the details of those enzymes and proteins. The first one is keto-acyl synthase. Keto-acyl synthase. It is called keto-acyl synthase. Let us give the abbreviation case. Okay. Then the next one is acetyl transacylase. Acetyl transacylase. It is AT. Okay. The third one is malonyl transacylase. Malonyl transacylase. It is called MT. The next one is dehydratase. Next one is dehydratase. We call it DH. Okay. Then the next one is enoyl reductase. It is called enoyl reductase. So it's a ER. The next enzyme is keto-acyl reductase. Okay. The another enzyme is, it is called KR. This is keto-acyl reductase is KR. Another enzyme is thioesterase. Okay. So it is, we can say this thioesterase TH. So thioesterase we can say or TE. It is TE. Okay. Then, so these are the various enzymes, and the protein present is acyl carrier protein. Protein is acyl carrier protein. Okay. These are the 7 enzymes and 1 protein. Now let me show how these enzymes, how these monomers, they are arranged. Okay. This is the how the monodimers are arranged. Let us show what are these various enzymes and proteins. This is, as I said, keto acyl synthase. This is acetyl transacylase. This is malonyl transacylase. This one is dehydratase. This one is enoyl reductase. This one is keto acyl reductase. Okay. Then this is ACP, acyl carrier protein. Okay. This is thioesterase. This is thioesterase. Like also we give here KS, acyl. Okay. So acetyl transacylase, malonyl transacylase, dehydratase, enoyl reductase, keto acyl reductase, ACP, and this one thioesterase. Okay, let us see how they are connected. These monomeric units, okay, how they are connected. Okay, so keto acyl synthase has got cysteine. It contains SH. Here it contains ACP contains 4-phosphopantethine. Okay, it contains SH group. So both are connected. These both are connected. Here also same thing. Here also it is same thing. Here 4-phosphopantethine. Okay, then here it is cysteine SH. Here SH. Both are connected through this one. Okay, this is the how these homodimers arranged in head to tail orientation. Okay, so this portion, when you see this portion, this one we can say divide into, this is the subunit division. Division. If you think in this format, it is active. No, it is not active. So there is a functional division is present. This is functional division. Is present. The functional division. So this one is one unit. This is one acts as one enzyme. So this is acting as the another part of the enzyme. That means this one can synthesize one fatty acid. This one is another fatty acid. See, all the enzymes are present in this unit. In this form, it is active. In this form only it acts. That means simultaneously fatty acid synthase multi-enzyme complex can synthesize two palmitic acids. Okay. What are the advantages? Of multi-enzyme complex. This multi-enzyme complex. There are many multi-enzyme complexes are there. Like pyruvate dehydrogenase multi-enzyme complex. Like alpha ketoglutarate dehydrogenase multi-enzyme complex. Like that also fatty acid synthase multi-enzyme complex. So this complex is associated with, okay, this is associated with, the particular complex is associated with, okay, specific metabolic pathway, okay, metabolic pathway, okay. So like, then alpha ketoglutarate dehydrogenase, so that is converting alpha ketoglutarate to the succinyl coenzyme. So like this, so the complex is associated with the one particular metabolic pathway. In this particular pathway, it is fatty acid synthesis. Okay, it is linked together. The enzymes are linked together. Okay, that is one advantage. The second advantage is several enzymes and also proteins packed together. They are packed together into one assembly. They are packed together into one assembly. That one important aspect is here, coded by. One important aspect is coded by a single gene. Okay, that is the important aspect. Several enzymes are packed together into one assembly and they are coded by a single gene. The third aspect is the most important is coordination. the maximum coordination is present when you see such multi-enzyme complexes. So among the enzymes, enzymes, in a series of reactions, so maximum coordination can be seen. The next advantage point is, such multi-enzyme complexes will lead to the higher catalytic rate. How it is possible? Let us explain. In this one, suppose there is a substrate. So it goes to the enzyme 1, substrate to enzyme 2, then intermediate goes to enzyme 3, so on, then ultimately product is released because all the enzymes are at one place. That is the reason the maximum efficiency will be there. The last point, but not the least, is maintains high speed. Maintains high speed. In where? In overall rate of production of final product. So if you compare with the reactions, individual enzyme reactions, the enzymes which take place in multi-enzyme complex, the product formation is very, very quick. So these are the different aspects of fatty acid synthase multi-enzyme complex. In this session, I have discussed about fatty acid synthase complex, which is required for the fatty acid synthesis. Let us summarize. This complex, the fatty acid synthase complex, it is a multi-enzyme complex. It is homodimer. 2 identical units are present. It catalyzes formation of 16-carbon palmitic acid. So as I said, the 2 monomers are there. They are identical, and each monomer contains 7 enzymes and 1 protein. How these monomers are arranged? The orientation is head to tail manner. The participating enzymes are keto-acyl synthase, that is KS, and acetyl transacylase AT, and malonyl transacylase MT, dehydratase DH, and enoyl reductase ER, ketoacyl reductase KR, and thioesterase TE. The protein is acyl carrier protein ACP. How these are arranged? So we will sum up in the form of a diagram. Let us see there are 7 enzymes. So here KS, AT, MT, and the another unit is DH, ER, KR, ACP, and thioesterase. They are arranged in this manner. So there at the ketoacyl synthase, cysteine acid is present. At the ACP of the other monomer, there is 4-phosphopantethene is present. The SH group is present. So both the monomers, they are linked through this SS bond. So this entire multi-enzyme complex can be placed as the subunit division and functional division. As subunit division, when you see it, it is not active. When you get this enzyme in the form of functional division, where we can say all the 7 enzymes, 1 protein is present, it is active. That means fatty acid synthase multi-enzyme complex can simultaneously produce 2 palmitic acid molecules. Advantages of multi-enzyme complex is the first one, associated with specific metabolic pathway linking all the enzyme reaction. The second advantage is several enzymes are packed together into one multi-enzyme complex coded by single gene. Then the third one is there is maximum coordination among enzymes in a series of reactions. Higher catalytic rate is ensured once substrate is taken up by the first enzyme, the intermediate goes to the second, third, and like that till the product is released. The reactions catalyzed by any multi-enzyme complex will have the high speed, and because of the maximum coordination, when all the enzymes are at one place, the substrate, once it enters, the final product comes out. These are various aspects of the features of multi-enzyme complex. Okay, thank you very much.